please revise. Don't keep anything as a backlog, huh? Because then it will be difficult for you all to ask me doubts. Okay, so we go ahead with biomolecules. Fine. Yeah, we had seen the structure of the purines and the pyrimidines and everything the other day, and today we're going ahead with the DNA, right? So we're going to talk about the structure of DNA. Now, as I said in the last class, that this is going to be a part which will be very helpful for you all in the study of genetics. So we have got chapters associated with genetics, which is going to be a syllabus for your 12th standard. So if you understand the DNA structure today and the RNA today, it will be like almost 30% of your portion is covered of your genetics in these particular sessions itself. So my dears, please pay very good attention to whatever is being said. So DNA is a long chain which is made up of alternate sugar and phosphate groups. So sugar, of course, is going to be your pentose sugar. Pentose sugar is the sugar which is having 5 carbon, correct? And it's going to have along with the sugar, it's going to have something called as the nitrogen bases, and along with the nitrogen base there is going to be a phosphate group. So we'll go in the details of all of these. So the sugar is always deoxyribose, and it is always joined to the phosphate in the same way, so that the long chain is perfectly regular, repeating the same phosphate sugar sequence over and over. इसका मतलब क्या होता है बेटा कि ये जो पेंटोज शुगर होता है, we have seen already carbohydrates, we have seen what is ribose sugar and deoxyribose sugar in that carbohydrate session I had told you, and I had also told you that the presence and absence of one oxygen molecule at the 2' end will decide the sugar to be deoxyribose or ribose sugar, correct? So the fifth carbon of the sugar will be always attached with the phosphate, okay, from the higher end. The next sugar, the next pentose sugar, okay, will also have a fifth carbon, है ना? दो sugars है, दोनों पे 5 carbon ये sugars है, दोनों पे है ना? दोनों 5 carbon sugars है. Now what will happen? There's always going to be a phosphate at the 5th carbon position. So remember this, that the sugar will be always present at the 5th carbon of the sugars. Phosphate group present होता है. But that phosphate group is going to be a connector between the two sugar molecules. That means the way the phosphate has attached at the 5th carbon, right? Similarly, ऊपर से भी वो किसी को अटैच होना चाहिए, है ना? तो ही वो कनेक्शन बनेगा ना. ऊपर के शुगर को और नीचे के शुगर को कनेक्ट करने के लिए there should be someone who will form common bridge, and this carbon which is having a phosphate at the 5th position, है ना, correct, will also get attached to the 3rd carbon of the first sugar. Now this is what is we have to understand very precisely. Okay, so I'll just show you with the help of diagram that how it looks like. It's very simple, it's very easy. Just see, each sugar of sugar phosphate chain has a base attached to it. Base मतलब कौन सा? Nitrogen base. Because it's DNA, because we're talking about deoxyribonucleotide, there are 4 bases: A, T, G, C. Adenine, thymine, guanine, cytosine. A, T, G, C. Okay, so each sugar will have phosphate at the 5th carbon, and it will have a nitrogen base, okay, attached at the first carbon. तो इमेजिन कर लो कि ये शुगर है, तो फिफ्थ कार्बन पे अगर फॉस्फेट है तो फर्स्ट कार्बन पे नाइट्रोजन बेस है. So could you now realize the sequence how it is going to be? Huh? Okay. So this unit, which consists of a sugar, phosphate and base, is called as a nucleotide. I guess we have already seen about the nucleoside and nucleotides. Correct? The nitrogen base and the sugar of a nucleotide forms a molecule which is going to be nothing but a nucleoside. And thus nucleoside does not contain a phosphate group. 4 types of nucleosides are found in DNA. In a nucleoside, nitrogen base is attached to the first carbon, as I said just now, of the sugar. When a phosphate group gets attached with the fifth carbon of the sugar, then that molecule will be called as nucleotide. तो साइड एंड टाइड में फ़र्क देखिए, मैंने आपको लास्ट टाइम भी एक शॉर्ट ट्रिक बताई थी कैसे याद रखना है साइड एंड टाइड को. डोंट गेट कंफ्यूज बिटवीन दैट. आई हैव ऑलरेडी गिवन यू दैट शॉर्ट ट्रिक, हाउ टू मेमोराइज और हाउ टू रिकलेक्ट. एग्जाम में कैसे मिस्टेक नहीं करेंगे हम. करेक्ट? Yeah, so these short tricks needs to be, like recollected. A single strand of DNA consists of several thousands of nucleotides, one above the other. So they are going to be stacked one above the other. How many? Almost several thousands. The phosphate group of the lower nucleotide is attached with the fifth carbon of the atom of the deoxyribose sugar and forms phosphodiester bond with that of the third carbon of the deoxyribose sugar of the nucleotide placed just above it. अब ये है ना लैंग्वेज बहुत ही ऐसे लगता है माय गॉड ये क्या है. It's okay. I'll come back to the language once I explain the diagram to you. Then it will be very very simple. Okay. Single long chain of polynucleotides of DNA consists of one end with sugar molecule, not connected with another nucleotide having carbon 3. Okay. Not connected with phosphate group. Similarly, the other end having C5 of the sugar is not connected with any other phosphate group. So they are going to be in a linear way. चेन होती है एक के ऊपर एक, और वो किसी से भी कहीं भी अटैच नहीं होते हैं. Okay? The two ends of the polynucleotides are called as 3' end and 5' end respectively. 3' और 3', 5' या 5'. ठीक है, ये देखिए. Now it will be easy for you all to understand what I am saying. Okay? This is your pentose sugar बेटा. ये है first carbon. Write it. See, once I write down, it will become all the more easy for you all. Here there is going to be your first carbon. Here you have got the second carbon. Here you have got the third carbon. Here you have got the fourth carbon, and this is going to be the fifth carbon. So 1 2 3 4 5 5 corners पे 5 कार्बन प्रेजेंट है. ठीक है. अब जो है look at this. Where did it go? So look at this. Here you have got the first carbon, second carbon, third carbon, fourth carbon, fifth carbon. Agreed? Now at the fifth carbon is present one phosphate group. Can you see? PO4 दिख रहा है आपको? Yes. अब ये phosphate जो है it is attached to the fifth carbon of the sugar जो नीचे की तरफ है. At the same time the same phosphate is also attached to the 3 carbon of the sugar which is present above it. So it's going to be a connector, है ना. तो phosphate ने थर्ड कार्बन के साथ भी बॉन्ड बना लिया जो उसके ऊपर है, और 5th कार्बन के साथ भी बॉन्ड बना लिया जो उसके नीचे है. Now understood what I'm trying to tell you? Correct? 3rd of the first and 5th of the second. Yes, exactly. But if you look at this one ना बच्चा, ये फर्स्ट शुगर है. तो ये जो फर्स्ट शुगर मॉलिक्यूल है इसके 3 प्राइम पे है लेकिन 5th प्राइम पे भी है. ये देखो 5 प्राइम पे भी फॉस्फेट अटैच्ड है. Okay, this is also going to be your phosphate, but this is going to be your only phosphate not connected to any 3 prime. Did you all understand? Okay, so this particular bond, only one with the phosphate, one single bond with the carbon is going to be called as phosphoester bond, while this phosphate which is connected to both the sugars, okay, is going to be called as phosphodiester bond. ठीक है? So please remember these. Why is the upper phosphate not connected to the 3rd carbon? Because probably there is no complementary base pair उसके सामने to connect with. It will only come, you know, when there is going to be complementary base pairs. So अगर adenine है तो thymine आएगा, guanine है तो cytosine आएगा. अभी इधर adenine और ये देखो बेटा, you can see here adenine and thymine. हाँ, but who will come here when there is no one over here? Okay. हाँ, so phosphoester will not get connected to anyone. It will remain as an ester bond and not diester. Di means 2. So when one phosphate molecule will get connected to 2 sugar molecules, one at 5 prime and one at 3 prime, only then we call it as what? Diester. Okay. So it is that the esters won't get connected anywhere. That is the reason we say that the 5 prime end of DNA is free. Free means there is no one ahead of it. Is it fine? Now understood this? Yeah, all of you my dears, if anyone is having any difficulty, please tell me. I'm again explaining it to you all one more time. Please pay attention. There is a phosphate attached to, हाँ मतलब PO4 है ना बेटा, that is PO4 ना. So that entire PO4 phosphate is coming and getting attached with your OH to form H2O. Okay, fine. So a water molecule is going to get eliminated even here. So in glycosidic linkages you saw for carbohydrates that the water molecule was getting eliminated. Similarly you saw when water molecule was getting eliminated during the peptide bomb formation as well. In the same way here also H and OH, okay, will be eliminated collectively as water molecule, and then that esters and diesters would be formed. Got it beta? Fine. So please pay attention one more time. This is your pentose sugar. Is there any change that the ester group might turn into if it comes in contact with some favorable compound? No my dears, no, because it's a proper coiled twisted DNA, so it's never going to be, you know, like reacting. Why the same can't happen for 3 prime end? 3 prime end के लिए भी सेम है ना बेटा. यहाँ पर अभी guanine and cytosine है, so if there is no one in front of it over here, who would come and get attached to it? No one can come and get attached to it. There should be what? Complementary pairing. A and T forms double bond. G and C forms triple bond. Where is it not connected? Here बेटा, इधर ऊपर वो किसी के साथ joint है क्या? किसी के साथ भी joint नहीं है ना. वैसे यहाँ पर वो किसी के साथ joint है क्या? किसी के साथ भी joint नहीं है. Okay. Which arrow is shown in the figure for the 3rd? I can't see him. This one, this arrow. It is just zooming that particular part and showing that what is exactly happening in that area. Okay, so यहाँ पर there is no one, here there is no one to combine with anyone else. Similarly, here there is no one to combine with anyone else. Got it? 3 prime end. Here, नीचे. This one you are talking about. So it is nothing, my dear. It is just to show that it is still, like only 2 have been shown. Na? So it's going to be a long chain of thousands. That is what they want to say. Phosphate is not shown attached to the 5, attached to. That is what, na beta, it is OH. So it's going to form a bond. Okay, it's going to form a bond with H2O. तो ये जो PO4 है ना, यही अटैच हो जाएगा with the carbon, that is CH2. Can you see? It is CH2. Correct? So OH and CH2 is going to be combining to form the elimination of water. Okay. So this is a very important point, my dears, which is going to really help you all in understanding the structure of DNA when we talk about DNA replication in molecular biology. That is your genetics. Any difficulty? Any problem? Here they are showing it as minus, my dear. So that itself will show it's going to be what? What do you mean by this minus sign? Can anyone tell me what is this minus sign? There is going to be a hydrogen bond between thymine and adenine. I am coming to it. There are 2 hydrogen bonds between thymine and adenine and 3 hydrogen bonds between guanine and cytosine. I am coming to it. What is this minus or negative sign over here? Can anyone tell me? Chemistry. Yes, come on. Oh, I'm getting so many answers. Anyone else who would give me an answer of this negative? Yeah, what is that electronegative charge? But ion क्या है? What is it? When we write it like this way, O minus. Come on. Chemistry है ये. It's not bio. Yeah, it's going to be phosphate ions in the ion form we are talking about. Correct? Right? So that is why they are showing it as a signature negative वाला. ठीक है, जैसे कि photosynthesis chapter में, respiration chapter में we have written it as NADPH plus H plus, if you know, if you recollect. Correct? So that was, yeah, so that is the way, like the way it's going to be written. So this is an important point, my dears, for DNA. So now once you have understood the structure, now come to the literature. The phosphate group of the lower nucleotide. See this. This is the lower nucleotide. Iska phosphate group. Huh? This one we are talking about. Ribose sugar. ये देखो. Is attached with the 5th carbon. इसका ये 5th carbon है ना? Correct? So this is the lower sugar. The phosphate is attached to the lower sugar ka 5 prime end. This is your 5 prime end. Okay. The phosphate group of lower nucleotide is attached to the 5th atom of the deoxyribose sugar to form phosphodiester bond with that of the 3rd carbon of the deoxyribose sugar of the nucleotide just placed above. So you can see over here, this is the 3 prime end. This is the 3 prime end. Correct? So from one side the phosphate is attached with the 5th carbon of the lower sugar. On the other side this phosphate is attached to the 3 prime of the higher sugar. So यहाँ पर एक एस्टर बॉन्ड बनेगा, यहाँ पर एक एस्टर बॉन्ड बनेगा, इसलिए दोनों जगह पर जो बनते हैं कलेक्टिवली हम उसको क्या कहते हैं? फॉस्फोडाइएस्टर बॉन्ड. It's nothing, my dear. It is just the ionic form of the phosphate. So it's going to be called as phosphate ion, which is going to come and establish this bond. Okay, that's the negative. Fine. Now you understood the language of it. That's why I said now once you understand the understand the diagram, it will be easy for you to look at the language. Okay, it's a single long chain of polynucleotide of DNA. One end with sugar molecules. One end of the sugar molecule means this one. This is one end of the sugar molecule, not connected to, see, not connected to another nucleotide. Okay, having carbon 3, C3 carbon, not connected with phosphate group. See, 3 carbon over here, not connected with any phosphate group. So first and the last will be alone. हाँ, so इसीलिए इसको हम कहते हैं 5 prime end, which is all alone, and this is called as your 3 prime end again, which is all alone. This is your opposite polarity होता है DNA का, इसलिए यहाँ पर 5 prime end, यहाँ पर 3 prime end. ठीक है. Similarly, the other end having C5 of the sugar is not connected with any more phosphate groups. So even here you can see the chain. This is 5 prime, this is 3 prime. Here if you see, this is 5 prime, this is 3 prime. Why do we call it as 3 prime? Look at this beta. This is first carbon, second carbon, third carbon, fourth carbon and the fifth carbon. So ये 5 prime end free हो गए. Okay. Similarly, this is first carbon, second carbon, third carbon, fourth carbon, fifth carbon. So here the 3 prime end is free, here the 5 prime end is free. Okay, now understood why we call it as 5 prime to 3 prime. Any difficulty in understanding any point, please let me know. All okay? Great. Yeah. The single polynucleotide strand of DNA is not straight, but it is helical. What do you mean by helical? It's going to be, you know, in a twisted form, the way you can see it over here now. So it's going to be helical, and it's not going to be a straight stacked one. Okay. The DNA molecule consists of 2 helical polynucleotide chains. Poly means many. Nucleotide का मतलब होता है sugar plus nitrogen base plus phosphate. Okay, so it's going to be a long chain of polynucleotide. Fine. The chains are complementary to each other. Now, what do you mean by the word complementary? See, the 2 complementary polynucleotide chains of DNA are held together by weak hydrogen bonds. So adenine will always pair with thymine, and guanine will always pair with cytosine. So purine and a pyrimidine will always be together. So adenine, thymine pair consists of 2 hydrogen bonds, and guanine and cytosine will have 3 hydrogen bonds. Okay. Thus, if the sequence of bases of one polynucleotide is known, the other chain sequence can be derived. You can determine the other chain. तो अगर यहाँ पर ऐसा chain है, as you can see over here very well, see here you can see it is A, तो you know here is going to be T. यहाँ पर G है तो here is going to be C. यहाँ पर T है तो here is going to be A. Okay, so this is the way, you know, you can determine the complementarity, because A will always pair with T and G will always pair with C, and vice versa, of course. Any difficulty in understanding these? Okay, now when we talk about the DNA model, this is the actual model which has been derived or which has been found out by scientists like Watson and Crick. So according to Watson and Crick, DNA molecule consists of 2 strands twisted around each other in the form of a double helix. So you saw just now like how they were bound together, right, in a double helix. The 2 strands, that is polynucleotide chains, are supposed to be in opposite direction. So end of one chain having 3 prime lies besides the 5 prime of the other. So here you can see this very clearly. यहाँ पर जहाँ पर 3 prime है उसके बिल्कुल बाजू में 5 prime है, और जहाँ पर 5 प्राइम है उसके बराबर सामने 3 प्राइम है. सो यू कैन सी हियर ऑल्सो दिस इज 5 प्राइम, दिस इज 3 प्राइम, दिस इज 5 प्राइम, दिस इज 3 प्राइम. व्हाट यू मीन बाय 5 and 3? वो समझा है क्या? ये इधर से कहानी चालू होती है. व्हाट एग्जैक्टली डू यू मीन बाय 5 प्राइम and 3 प्राइम? एनी डिफिकल्टी? नहीं तो पूरी कहानी सुनोगे और बोलोगे उसको 5 प्राइम और 3 प्राइम क्यों कहते हैं लेकिन. हाँ, इज इट लाइक दैट वे? और यू ऑल नो what exactly is 5 prime and 3 prime? No, not because it's a double helix. The reason is the 5th carbon is free and the 3rd carbon is free of the first and the last sugar respectively. Yes, okay. So in one chain the 5th carbon is free, not connected with anyone. I mean the phosphate not connected with anyone, and the 3 carbon OH is free, not connected to anyone. So 5 and 3 denotes the position of the carbons which are free. ठीक है. हाँ, अदरवाइज इधर से फिर ये क्या चल रहा है, ये 5 प्राइम क्या है, 3 प्राइम क्या है, कुछ नहीं समझ में आ रहा है. जिनको नहीं समझ में आ रहा है वापस एक बार बता रही हूँ बेटा. प्राइम और डैश इज यूज्ड टू डिनोट the position of the carbon of a sugar. Okay, carbon of a sugar is denoted by a prime, dash, etc. prime, it's called as dash or prime. Okay, fine. So when I say 5 prime means the 5th carbon of the pentose sugar is going to be free, not connected to any other nucleotide. When I say 3 dash, the 3rd carbon of the pentose sugar is free, not connected to any other phosphate. Is it fine? ये यहाँ से सबसे इम्पोर्टेंट कहानी यहाँ से स्टार्ट होती है अपनी. Well, so the 2 strands of the polynucleotide chains are opposite. So the chain having 3 prime lies just beside with the 5 prime end. One turn of a double helix of DNA measures about 34 angstroms. A and उसके ऊपर ये डिग्री वाला एक ये दिख रहा है आपको symbol. How we write Celsius degrees वैसे. So that A stands for angstrom. Well, so one turn of the double helix. Now what do you mean by one turn? ओके, सो फ्रॉम हियर टू हियर एंड, ये देखो ये एक टर्न है. तो मेरे दोनों हाथ के ऊपर अगर आप फोकस करोगे तो वन इज फेसिंग दिस साइड, वन इज फेसिंग दिस साइड. कैन यू सी द वे आई हैव हेल्ड इट? सो फ्रॉम दिस एंड टू दिस एंड, ओके, फ्रॉम माई दिस थंब टू माई लिटिल फिंगर, दिस इज वन टर्न बेटा. बहुत बार बच्चों को लगता है सिर्फ ये एक है, ये एक टर्न है. नो, दिस इज वन टर्न. So in the diagram over here you can see one turn क्या होता है. I'll show you one turn over here. See, so from this end to this end is one turn. Did you all understand? तो turn मतलब ये turn की बात नहीं होती है. Turn is from this to this. When you move your hand, one turn. Please, huh? This is an important point. Okay, yeah. So they are saying that this is going to be one turn, and one turn of the double helix of the DNA measures about 34 angstroms, also called as 3.4 nanometers. So you know the conversion now. 3.4 nanometers, also called as 34 angstroms. It's one and the same. Okay. It consists of paired nucleotides, that is AT and GC, and the distance between the 2 neighboring pair of the nucleotides is 3.4 angstrom या 0.34 nanometers. इससे क्या पता चला बेटा? इससे क्या पता चला कि the distance between 2 अगर 3.4 है और total length अगर 34 है तो 34 divided by 3.4 10 आ गया. So there have to be 10 nucleotide bases present in that turn. करेक्ट। एक टर्न में 10 न्यूक्लियोटाइड बेस पेयर्स होते हैं, तो AT GC 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 AT AT TA GC कुछ भी हो सकता है, but they have to be 10 in one particular turn. So let us see if they are 10. देखिए, let us count. See, 1 2 3 4 5 इसके अंदर छिप गया है, 6 7 8 9 10 यहाँ पर छिप गया है. So there are 10 base pairs present between this one turn. Okay, and the distance is how much? 3.4 nanometers. Distance between two consecutive is going to be 0.34 nanometers. समझा? Okay. Order doesn't matter. Yeah, order doesn't matter, beta. It could be anything. हाँ, the number should be 10. Yes. The diameter of the DNA molecule has found to be 20 angstroms. There are certain organisms like bacteriophages, like 5X174, and several bacterial viruses which possesses a single-stranded DNA. So if there is a single-stranded DNA, there is no question of making pairs. So bacteriophage, of course, is the virus which is going to attack the bacterial cells. Correct? So they are going to be single-stranded DNA. So they don't form base pairs. But in us, base pairs. Any point not followed, not understood of this which is there on your screen right now? These questions are very frequently asked in your exams. What are the distances and how many turns and all those things. All okay? One turn? I'll show you again. This is your one turn. From this to this is going to be the one turn. Okay. यहाँ से लेकर यहाँ तक 10 हमने काउंट किए ना, 1 2 3 4 5 इसके अंदर, 6 7 8 9 10 इसके अंदर. तो from this to this is going to be one turn. Okay. In a turn there are 2 pentose sugars. No. There are 10 pentose sugars. One pentose sugar will have one. So one on one strand. So इसके साथ एक पेंटोज, इसके साथ दूसरा, तीसरा, चौथा, ऐसे 10 पेंटोज शुगर इधर, 10 पेंटोज शुगर इधर. Okay? Fine? Please understand. Bacteriophages will have single-stranded DNA. The meaning of that is simple. That bacteriophage 5X174, that is the name of that bacteriophage, will have a single-stranded DNA. They don't form double-stranded DNA. ठीक है? Got it? Last point. Got it now? It's going to have a single-stranded DNA. There are many organisms with single-stranded DNA as well. That's all with your DNA that you have to understand. Any difficulty? Can I move to RNA? Okay. Another nucleic acid found in living organisms is the ribonucleic acid. In most of the organisms, it is not found to be hereditary material. It's a nucleic acid. Ribonucleic acid is going to be, RNA is going to be a nucleic acid, no doubt. But it is not the hereditary material in majority of the cells. Okay? Though in certain organisms like tobacco mosaic virus, it is the hereditary material. So what do you mean by hereditary material? The ones which is going to be passed on from one generation to the next, carrying the genetic information of that particular organism. So even in us, RNA is not playing any role with heredity. It's continuously the DNA which is going to be the genetic material, which is going to have all the information about the different characters that we have got. But we'll have RNA. Without RNA, protein synthesis होगा ही नहीं बेटा. तो हमारे में हमारे सेल्स में आरएनए का बहुत बड़ा प्राइम रोल है. दैट इज इट इज हेल्पिंग इन प्रोटीन सिंथेसिस. ठीक है. So see, like DNA, ribonucleic acid also consists of polynucleotide chain with the difference that it consists of single strand. So generally, in we all, RNA will not be double stranded, and precisely not the way it is in DNA. Even if it is double stranded, it's not. It's going to be looped, but not helical. ठीक है. Example: reovirus, wound tumor virus. Okay. RNA is double stranded in these cases. The nucleotides of RNA have ribose sugar instead of deoxyribose sugar, as in case of DNA. So I think majority of you all remember the difference between RNA, the ribose sugar and the deoxyribose sugar. Do you remember the difference? Can anyone tell me that in DNA, which position has got one oxygen less? Which carbon has got one oxygen less? Come on, who will tell me? RNA पे सब जगह पर OH है, DNA के एक जगह पर H है, O नहीं है. तो which is that position of the carbon where O is absent? 1 2 3 4 5 कार्बन तो है. उसमें से कौन से इसमें? वेरी गुड भूमि, राइट. Yes, Jaya, correct. Come on, others. Good morning. Come on. Only 2 of them have answered till now. Others. Yeah, Jaya is right and Bhumi is right. Okay, Siddhika, right. Come on, others. All of you. हाँ, कितना सिंपल क्वेश्चन है ये. Yes, Ayush, you are right. Okay, Zui, why a question mark? You are right, my dear. It's at the second position. 2' end of your sugar, ribose sugar के second position पे OH की जगह पर अगर H है, then it will be called as deoxyribose sugar. Oh my god, I think I have to go back with your carbohydrates, right? We had done this. I had drawn this diagram also. Even in nucleotides, I guess I had done this. मैंने ड्रॉ भी किया था बोर्ड पे ये पूरा. हाँ. Yes. Two prime. 2' पे नहीं होता oxygen. I had drawn this also in the class if I'm not wrong. Okay, those who are confused उनके लिए है ये. This is your first carbon, second carbon, third carbon, fourth carbon, fifth carbon, CH2HOH. Okay, at the second position you have got OH and H. Here also you have got OH and H. Okay, correct. अब ये जो है this particular diagram signifies what? Either is one OH, here there is one more OH. On the other hand, if I draw this diagram exactly the same as I have drawn, no difference. Can you see here? So this is your first carbon, this is second, third, fourth and fifth. So here you can see on the second carbon there is one oxygen missing, one oxygen less, इसीलिए इसको हम क्या कहते हैं, deoxy, and this is your ribo, this is deoxy ribo. समझा? जिनको confusion था कि what I'm talking about. So this is your pentose sugar called as deoxyribose if one oxygen is less at the second position of the sugar, and if it is having OH then it will be called as your ribose sugar. Is it fine with all of you? Still any confusion in your minds? Done? So that is what they are talking about. The nucleotides of RNA have ribose sugar instead of deoxyribose sugar as in case of DNA. So you understood what I am trying to tell you. In case of RNA, uracil substitutes thymine of DNA. So ATGC was a part of DNA, while AUGC will be a part of RNA. So RNA में थाइमीन नहीं होता है, RNA में यूरासिल होता है. सिमिलरली DNA में यूरासिल नहीं होता है, DNA में थाइमीन होता है. ये बेसिक चीज़ें हैं, वेरी इंपॉर्टेंट. ओके. Purine pyrimidine equality is not followed in RNA molecule because of its single stranded structure. So it's not a rule that जितने प्यूरीन है. अच्छा, I hope you all know what are purines and what are pyrimidines. ये भी हमने किया था, but still if you are confused. Purines are double ring structure. Pyrimidines are single ring structures. है ना? Yes. So your pyrimidines are going to be thymine, cytosine and uracil. Okay. And your purines are adenine and guanine. Yes. Okay. So RNA strand is usually found folded upon itself in certain regions or entirely. These folding helps in stability of the RNA molecule. Most of the RNA polynucleotide chains start either with adenine or guanine. There are 3 types of RNAs which have been found in our cell cytoplasm. So we call them as cellular RNA, not in the nucleus. Okay. That is going to be your mRNA, messenger RNA, or we also call it as template RNA for protein synthesis, rRNA or ribosomal RNA, and tRNA or transfer RNA. It's also called as soluble RNA. Okay, so what are the differences between these 3 is something that we have to do into details. First, I talk about the mRNA. mRNA carries genetic information for arranging amino acids in definite sequence. So what is exactly going to happen, you know? The DNA will have suppose, okay? I'll just explain. It's just suppose, okay? So suppose we have got DNA. This is your 5' to 3' DNA, and of course this is going to be your 3' to 5'. Opposite directions में होते हैं बेटा DNA के strands. हाँ, अब देखो यहाँ पर है A A T T A G G C A C. फॉर एग्जाम्पल बता रही हूँ मैं. तो इसके कॉम्प्लीमेंट्री स्ट्रैंड पे क्या होगा बेटा? A will pair with T T A A T C C G T G. Correct? They are complementary. A pairs with T, T pairs with A, G pairs with C, C pairs with G. Correct? Right? Now what happens is this mRNA molecule will be, I'll show you, this mRNA molecule will be synthesized on one of the strands of DNA. अब इसका मतलब क्या होता है पता है? डीएनए के पास जो इंफॉर्मेशन है AATT AGGC AC whatever it is will be copied as mRNA. जैसे तुम्हारे पास ओरिजिनल डॉक्यूमेंट्स होते हैं कि नहीं? तुम्हारा आधार कार्ड है, तुम्हारा आधार कार्ड ओरिजिनल है, तुम्हें उसके कॉपीज निकालने हैं, तो व्हाट यू विल डू, यू विल टेक आउट जेरोक्स कॉपीज, करेक्ट? तो वो जो जेरोक्स निकल रहा है ना, इट इज व्हाट, it is the same replica of the original. Do you agree to this? Correct? But the paper is different. Paper is different. In the same way, my dear, this particular mRNA will copy this strand but from this strand. इसका मतलब समझ रहे हो? ये देखो A I said कि RNA में T नहीं होता है, तो this A will copy what? A G G C A C. Agreed? But the way I have written, you can see कि मैंने 3 प्राइम वाले का टेम्पलेट यूज किया और 5 प्राइम टू 3 प्राइम मैंने mRNA बनाया. अब अगर देखोगे तुम, तो सी ये सेम टू सेम ए एंड ए सेम कॉपी आ गई, ए एंड ए सेम कॉपी आ गई. हाँ, टी के जगह पर यू आ गया, ए एंड ए सेम कॉपी, जी एंड जी, सी एंड सी, ए एंड ए, सी एंड सी. हाँ, ओनली इंस्टेड ऑफ टी यू रहेगा, रेस्ट एवरीथिंग विल बी कॉपीड एज इट इज. ओके, got it? So this yellow sequence that I have made now, this is my yellow sequence which I have made from 5 prime to 3 prime. That is what? A U A G G C A C. Do you agree? समझा क्या हो गया? तो ये जो 5 प्राइम टू 3 प्राइम येलो कलर का मैंने स्ट्रैंड बनाया है ना, बिकॉज इट इज हैविंग सी यू एंड यू, दिस इज गोइंग टू बी योर आरएनए स्ट्रैंड कहाँ से कॉपी हुआ, कहाँ से उसने जेरोक्स ले ली? फ्रॉम द डीएनए, करेक्ट? आरएनए ने जेरोक्स कहाँ से ले ली बेटा? डीएनए से ले ली कि नहीं? So the information which is present in the DNA will be copied onto mRNA. Did you understand? So DNA to mRNA, the process is going to be called as transcription. White is DNA, yes, and yellow is RNA. Exactly बेटा. समझाना क्या हुआ? किसी को डाउट है, कुछ डिफिकल्टी है समझने में? तो इसने बराबर AAU कैसे लेके आए? How is it possible to get the perfect nucleotides? It is possible to get the perfect nucleotides because of the template. Template समझते हो? What is a template? A mold. Mold पता है? हार्ट शेप का केक होता है, तो वो हार्ट शेप का मोल्ड होता है ना? स्फेरिकल केक बनता है तो स्फेरिकल शेप होता है. ओके, एनी वन स्ट्रैंड ऑफ डीएनए इज टेकन हियर फॉर ट्रांसक्रिप्शन. इट कैन बी रैंडम बेटा. ऑलवेज रिमेंबर दैट फॉर ट्रांसक्रिप्शन ऑलवेज द डीएनए टेम्पलेट इज टेकन फ्रॉम 3 prime end to 5 prime end because any strand to be synthesized should be from 5 to 3. ये डायरेक्शन हमेशा याद रखिएगा. कोई भी सिंथेसिस जो है वो 5 प्राइम टू 3 प्राइम होता है, कभी भी 3 प्राइम टू 5 प्राइम सिंथेसिस नहीं होगा. तो one train going to Churchgate, the other train going to Borivali. Okay, got it? That should be the way. So any new synthesis, let it be DNA synthesis, let it be RNA synthesis, new strand formation shall always take place from 5 prime to 3 prime. And we know that 5 prime to 3 prime अगर हमें चलना है तो एंटी सेंस होना चाहिए, उसके उल्टा वाला स्ट्रैंड हमें देखना है. So always the template will be what? 3 to 5. Yes. Okay, got it? So this yellow one is going to be your mRNA and this process is going to be called as transcription. So ये जो है ना, ये वाला जो strand है, ये वाला, this one, this will be called as your template strand. इसीलिए टेम्पलेट का कॉम्प्लीमेंट्री पेयर आ रहा है. अगर टेम्पलेट पे T है तो कॉम्प्लीमेंट्री A आया, टेम्पलेट पे A है तो कॉम्प्लीमेंट्री U आया, टेम्पलेट पे G है तो कॉम्प्लीमेंट्री C आया. सो लॉजिकली अगर देखोगे तो इसके कॉम्प्लीमेंट्री पेयर्स जो mRNA पे है वो सेम सीक्वेंस 5 प्राइम टू 3 प्राइम DNA पे है. Yes, always 5 prime to 3 prime DNA. Yes. अब कुछ चमक रहा है, आप कुछ समझ में आ रहा है कितना ब्यूटीफुल मैकेनिज्म है ये. Okay, so the DNA strands are, and let me tell you, the entire DNA is not going to be copied as mRNA. Okay, some areas of DNA will be copied as mRNA. Which are those some areas? Molecular basis of inheritance. I'll explain you during that time, of course. U, A, C, G, yes, correct. RNA would be 3 to 5 नहीं, RNA is also 5 to 3. ये देखो 5 to 3. Okay. So what is going to be copied? DNA strand will be copied. Which DNA strand would be copied? The DNA strand having a polarity from 3 to 5 will be copied as 5 to 3 of RNA. ठीक है? Clear? समझा? हाँ, सबको समझा? That is the reason I said, my dears, 30% of your genetics आज के लेक्चर में कंप्लीट हो जाएगा. अच्छी तरह से ध्यान दोगे तो. Don't get confused. If there is any confusion, please feel free to ask. Okay, it's very crucial part of our syllabus is going on. This thing. Fine. Let's go ahead. Okay. So that's what they are trying to tell you all. So mRNA carries genetic information for arranging amino acids in definite sequence. It's a linear polynucleotide. See, it's 5 prime to 3 prime. This is your mRNA. It accounts for only 3% of the cellular RNA. Its molecular weight is several millions. mRNA molecule carrying information to form a complete polypeptide chain is called as a cistron. So that's what I said, that entire DNA will not be copied. Only a part of DNA will be copied. So that part of DNA which is going to be copied as mRNA will be called as a cistron. Okay. Size of mRNA is related to the size of message it contains. So if there are more nucleotides which have been copied, then the mRNA size will be longer. If less nucleotides have been copied, it will be shorter. Synthesis of mRNA begins at 5 prime end of the DNA and terminates at the 3 prime end of the DNA. Okay? So it's always from 5 prime to 3 prime. Please remember this. Anything that is going to be synthesized, let it be DNA synthesis, let it be RNA synthesis, it is always going to take place from 5 prime to 3 prime. Got it? This is your mRNA. Look at the rRNA. rRNA forms 50 to 60% of the ribosomes. Okay? It accounts for 80 to 90% of the cellular RNA. So your mRNA was only 3%, whereas rRNA is almost 90%. It is synthesized in the nucleus. Rather, I would say it is in the nucleolus. Nucleus के अंदर जो nucleolus है, nucleolus is the factory for rRNA synthesis. ठीक है. Kurland discovered rRNA. It gets coiled here and there due to interchain complementary base pairing. इसका मतलब क्या है? By chance ATGC आमने सामने आ गए तो डबल स्ट्रैंड्स ऐसे बन सकते हैं, by chance. But mRNA is always a linear molecule. This is a thumb rule. mRNA will never fold. mRNA is going to be a straight linear molecule. rRNA might fold to form loops here and there. Okay, look at this. Most important, tRNA. Why am I saying most important? It is most important. Genuinely बोल रही हूँ, बहुत important है ये. tRNA molecules are much smaller, consisting of only 70 to 80 nucleotides. Fine. It is also single-stranded, but a number of complementary base sequences are going to be formed after pairing. It is going to be appearing like a clover leaf like shape. तो clover leaf पता है? ये ऐसे कुछ clover leaf के जैसा दिखता है ये वाला tRNA का shape. Hawley had proposed this clover leaf shape. Each tRNA can pick up a particular amino acid. Now my dears, this is again a very very important point. Okay, what happens is, this is going to be your tRNA. Here you can see 5 prime. Look the way my cursor is moving. 5 prime is moving like this way and it is coming to your 3 prime. So the two ends 5 prime and 3 prime. Can you understand how we have gone? So 5 प्राइम से हमने स्टार्ट किया, ऐसे ये स्ट्रैंड बनता गया, लूप्स होते गए एंड फाइनली ये 3 प्राइम एंड पे एंड हुआ. एट दिस 3 प्राइम एंड देयर इज गोइंग टू बी एन अमीनो एसिड अटैचमेंट साइट. व्हाट इज द नेम? अमीनो एसिड अटैचमेंट साइट. कहाँ पर है? 3 प्राइम एंड पे है. ठीक है. अब ये कैसे पिकअप करेगा? ये अमीनो एसिड जो है ये कैसे पिकअप करेगा? So the amino acid is going to be present in our cytosol. Whatever we eat as proteins will be broken down into amino acids. These amino acids will remain in your cell cytoplasm. And these amino acids will be double seat लेगा ये. आजा मेरे गाड़ी में बैठ जा. कौन बोलेगा? Who will say this? आजा मेरी गाड़ी में बैठ जा. कौन बोलेगा? The 3 prime end of the tRNA. किसको बोलेगा? अमीनो एसिड को बोलेगा. कौन से अमीनो एसिड को बोलेगा? किसी भी अमीनो एसिड को पिकअप करके लेके आएगा क्या? Never. So what decide that which amino acid should go and sit on that 3 prime end? So that particular thing, कि which amino acid should go and sit on that particular end will be decided by this sequence called as anticodon. What we call it as? Anticodon. Now what do you mean by this anti? Anti means against. Codon क्या होता है पहले वो समझना पड़ेगा अपने को. So codon is a triplet of nucleotide present on mRNA. What is codon? A triplet of nucleotide present on mRNA. तो जैसे अगर आपको याद है AAC GGU जो भी हमने लिखा था. Okay, so AAC is one triplet, UUG another triplet, CGA one more triplet. तो जो अपना mRNA का सीक्वेंस होता है ना बेटा, हाँ, it is read as a triplet. तो अगर फॉर एग्जाम्पल हमारे mRNA पे है ये ट्रिपलेट, सो अगर हमारे mRNA पे ये ट्रिपलेट है 5 prime 3 prime. Suppose we have got AUG CCC AAG whatever. Okay, GGU ये अगर ट्रिपलेट्स है तो यहाँ पर क्या है? कैन यू सी AUG दिखाई दे रहा है आपको? AUG एज वन ट्रिपलेट. तो द एंटीकोडॉन विल हैव व्हाट यू नो, UAC. Now don't you think A will pair with U, U will pair with A, G will pair with C. तो कोडॉन का कौन सा सीक्वेंस है, उसके कॉम्प्लीमेंट्री सीक्वेंस होते हैं एंटीकोडॉन पे. समझा कि नहीं समझा? अगर C है कोडॉन पे, तो बताइए मुझे एंटीकोडॉन पे कौन सा सीक्वेंस रहेगा. कम ऑन टेल मी. इफ द कोडॉन इज C. वेरी गुड. यस, आई वांट ऑल ऑफ यू ऑल टू आंसर दिस क्वेश्चन. वेरी गुड, वेरी गुड, वेरी गुड. So if the codon is C, of course the anticodon will be G. Correct? If the codon is AAG, come on tell me what will be the anticodon. AAG का anticodon. Very good, very good, very good. All of you all are getting. Oh my dear, it is not T नहीं रहेगा, T नहीं रहेगा. ये mRNA है, rRNA है. RNA में T नहीं होता है. क्या आ जाएगा इंस्टेड ऑफ टी? व्हाट इट वुड बी? इंस्टेड ऑफ थायमीन व्हाट इज गोइंग टू बी प्रेजेंट माय डियर्स? हाँ, नाउ यू ऑल गॉट इट. सो डोंट मेक दिस ब्लंडर. ओके, सो हियर इट इज गोइंग टू बी UUC. चलो GGU के लिए बोलो. लास्ट है GGU. कम ऑन, व्हाट वुड बी द एंटीकोडॉन? वेरी गुड, वेरी गुड. यस. कृष्णा, एक बार देख ले बेटा. G का कॉम्प्लीमेंट्री क्या होगा? G विल पेयर विथ व्हाट माय डियर? Guanine. हाँ, so your answer will be CCA. Correct? Understood this? Now, what do you mean by this? I'll tell you the meaning of this also. AUG अगर कोडॉन पे है, तो एंटीकोडॉन UAC एकदम मतलब एकदम खुश हो जाते हैं. अरे यार मेरा नंबर लग गया. एंटीकोडॉन UAC खुश हो जाता है कि मेरा नंबर लग गया. मेरा नंबर क्यों लग गया? ताकि मैं डबल सीट ले लूँ अमीनो एसिड को. सो जैसे ही कोडॉन आ जाते हैं, इमीडिएटली कॉम्प्लीमेंट्री एंटीकोडॉन खुश हो जाते हैं. तो जो AUG है ना बेटा, वो अमीनो एसिड मेथियोनिन के लिए कोड करता है. इसका मतलब क्या हो गया? AUG आया mRNA पे, हाँ, एंटीकोडॉन UAC गॉट एक्टिवेटेड. UAC वेंट एंड टोल्ड द मेथियोनिन, आजा मेरी गाड़ी में बैठ जा. Got it? So this amino acid will be your methionine, who will come and sit on this particular double seat on the 3' end. Now what will happen? Now you understood the connectivity, how the protein synthesis is going to initiate. So this is one amino acid which will come. So AUG UAC को सिटी मारता है, UAC methionine को सिटी मारता है. ओके, गॉट इट. वैसे CCC किसको कोड करता है? कौन से अमीनो एसिड को? AAG कौन से अमीनो एसिड को कोड करता है? GGU कौन से अमीनो एसिड को कोड करता है? दिस इज द ब्यूटिफुल स्टोरी दैट वी विल बी स्टडीइंग इन मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस. एंड वी विल ऑल्सो सी कि एक के पीछे एक ये अमीनो एसिड की चेन कैसे बनती जाती है, जिसको हम पॉलीपेप्टाइड कहते हैं, which is nothing but your linear protein which will be folded to form your 2-dimensional, 3-dimensional, 4-dimensional structures of the proteins eventually. Understood? So ये जो लूप है एंटीकोडॉन, ये जो लूप है, it gets, okay, it's going to have these specific sequences on them. तो जो कोडॉन है कॉम्प्लीमेंट्री एंटीकोडॉन का गुड मॉर्निंग हो जाता है. थोड़ा समझ में आया ये प्रोटीन सिंथेसिस का कहानी. सो अगर RNA नहीं होता है ना बेटा, देन it's not possible for proteins to get synthesized. It's very important for the proteins to get synthesized, right? It's a very beautiful journey, very beautiful journey. and of course I'll be dealing with it eventually. हमें करना ही है ये. We have to go ahead with the syllabus with molecular basis. Anyways, so see, each tRNA can pick up a particular amino acid. Now you know that how that particular amino acid will be picked up, right? Following 4 paths can be recognized on tRNA. One will be called as your DHU loop. This is your DHU loop. That is dihydroxy uracil loop, also called as amino acid recognition site. Second will be amino acid binding site. Third will be your anticodon loop. And fourth will be your ribosome recognition site. So this is going to be your T loop, is your ribosome recognition site. What is it? I'll explain when I'll explain your protein synthesis. तो 4 loops याद रखिएगा बेटा. DHU, anticodon, T-side and amino acid binding. ठीक है. ये जो है ना, this is called as T-side loop also. ये साई है बीच में symbol. In the anticodon loop of tRNA, 3 unpaired nucleotides are present called as anticodon, which pair with the codon present on mRNA. So if it is AUG on the mRNA, UAC will be the anticodon which will form a pair, and so on. The specific amino acid is attached at the 3' end in the acceptor stem of the chloroleaf of tRNA. Now you know which is that specific amino acid. So the amino acid will be decided by the sequence of codon. Okay? Between 5 and 3. This is a variable loop. Till today we don't know what is the function of this variable loop. ये जो है ना, ये वाला, इसको हम variable loop कहते हैं. But हमें ही पता नहीं अभी तक इसका function क्या है variable loop का. Okay? Well, so that's about your tRNA. So you have understood the nucleic acids very precisely, beta. Any difficulty, any problem in understanding any part of your nucleic acid, that is DNA and RNA? किसी को कोई difficulty हो तो please ask me. A pairs with T in case of DNA and with U in case of RNA. DNA में AT, RNA में AU. Clear? Okay. Then we go ahead with the lipids. Lipids इतना simple है, it's not difficult at all. They are generally water insoluble. We all know that lipids are water insoluble. Okay? Yeah. So in lipids, hydrogen to oxygen ratio is greater than 2:1. In carbohydrates, it was 2 parts of hydrogen and 1 part of oxygen. In lipids, it's going to be more than 2:1. They could be simple fatty acids. A fatty acid will have what? Acid है तो carboxyl group COOH, which will be attached to the R group. The R group could be methyl, ethyl, or a higher number of CH2 groups from 1 to 90. कुछ भी हो सकता है. Okay? The 5' end shown in the previous diagram is of tRNA or tRNA. It's the tRNA. Yeah. So the R group could be anything between 1 to 19 carbon. Okay. For example, palmitic acid has 16 carbon, including the carboxyl carbon. Now this is important. Total 16, including COOH वाला C भी इसमें included है. Now this is a question that they had asked 3 years before. Okay, 16 carbon वाले को क्या कहते हैं, 20 carbon वाले को क्या कहते हैं. तो देखो, it is arachidonic acid, which is going to have 20 carbon atoms including the carboxyl group. Okay, fatty acids could be saturated with double bond or unsaturated with 1 or more than CC double bond present. Another simple lipid is glycerol, which is trihydroxypropane. What is glycerol? Glycerol is trihydroxypropane. These points they ask you in your exams very frequently. Many lipids have got both glycerol molecule attached with the fatty acids. So here the fatty acids are found esterified with glycerol. Okay, ester bonds बनते हैं इनमें. They can be monoglycerides, diglycerides, triglycerides. We are always tensed about our triglycerides, right? Blood में triglycerides बढ़ने नहीं चाहिए is what the doctor will always tell you. So glycerol plus single fatty acid, 2 fatty acids, 3 fatty acids, monoglyceride, diglyceride and triglycerides. These are also called as fats and oils based on melting point. Okay, so oils have lower melting point. We know this. Example, gingerly oil, and hence remain as oil in winters also. So even we consume our oils, na, which will remain as oil only, liquid only. Correct? Well, some lipids have phosphorus and phosphorylated organic compound in them. This will be called as phospholipids. So we know the cell membrane is made up of phospholipids. Our cell membrane, correct? They are found in cell membrane. Lecithin is one example of phospholipids. Some tissues, especially the neural tissues, have lipids with more complex structures. So the nervous tissue, we know myelinated sheath, non-myelinated sheath. Do you remember all these? So obviously they are going to be what? Lipids. Okay, look at this unsaturated fatty acids and saturated ones. The difference between them is very clearly seen. This is your unsaturated fatty acid. So unsaturated fatty acids are with one or more double bonds between the carbon atoms of the hydrocarbon chains. So this is one hydrocarbon chain, this is another, and you have got a double bond, carbon-carbon between. Correct? Oleic acid found in nearly all fats and linoleic acid found in many seed oils are examples of unsaturated fatty acids. Remember the examples of unsaturated: oleic and linoleic. ठीक है? These fatty acids are basic molecules which form different kinds of lipids. Lipids may be classified as simple lipids, compound lipids, or derived lipids. Okay? Look at this saturated fatty acid, but there is no double bond carbon anywhere over here. Let us go to the simple lipids. These are esters of fatty acids with various alcohols. So they are going to have an alcohol tail. Okay. Fats and waxes are simple lipids. Fats are esters of fatty acids with glycerol. CH2OH, CH, CH2OH, CH. ऐसा उसका chain होता है. Triglycerides are 3 molecules of fatty acids which will be esterified to 1 molecule of glycerol. Okay. Generally, unsaturated fats are liquid at room temperature and are called as oils. Unsaturated fatty acids are hydrogenated to produce fats. For example, वनस्पति घी. तो जो वो वनस्पति घी आजकल पता नहीं, मतलब I don't know if you all use it. Generally, we don't use वनस्पति घी, but the best example of वनस्पति घी is डालडा. Yes, डालडा जो मिलता है ना मार्केट में, उसमें ऑयल्स में हाइड्रोजनेशन कराकर ओके उसको घी में कन्वर्ट किया जाता है. डालडा जो मार्केट में मिलता है. Fats are nutritional sources with high calorific values. So they are going to be actually very good energy sources. Fats act as reserve food materials. In plants, it is stored in seeds to nourish embryo during germination. And that is the reason that many seeds are oil seeds. हम उसमें से oil करते हैं. तिल का तेल, मूंगफली का तेल खाते हैं कि नहीं? सैफ्लावर, सनफ्लावर, हाँ, और क्या बताया आपको? राइस ब्रान. What all oils do you know? Coconut oil, how can we forget? So how come we get oil from them? So the plants are going to, sunflower, safflower, exactly. So the plants are going to store these. Even olive oil. How can we forget olive oil? हाँ, पास्ता खाते हो कि नहीं? Continental food. All continental food is made in olive oils. Do you know this? Yes. So, yeah, of course. But we are here. Yes, of course. Fish oil भी होता है. Cod liver oil. Heard about this ना? Cod liver oil. Yes, 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 yes. You are right, Bhubi. Yeah. So we have got different types of oils. कहाँ से आया oil वहाँ पर? These are going to be the plants which are going to store the fats in the form of these oils. Okay. In animals, fat is stored in adipocytes of the adipose tissue. We have done structural organization. We know what is adipose tissue. White fat, brown fat, सब हमने डिटेल में किया था. Fats deposited in subcutaneous tissue act as insulator and minimize loss of body heat. So they act as insulators. Subcutaneous means below the skin. Correct? Fats deposited around the internal organ act as cushions to absorb mechanical shock. Yeah. So even our visceral body organs are going to be surrounded by fatty layers. The main intention is taking care of those delicate organs. Okay. Wax is another example of simple lipid. They are esters of long chain fatty acids with long chain alcohols. They are found in blood, gonads, sebaceous glands of the skin. हर जगह पर तुम्हें ये fats मिलते जाएंगे. Waxes are not readily hydrolyzed as fats and are solid at ordinary temperatures. For example, waxes in beehive. This beeswax is used in cosmetics. पता है आपको? Girls, I don't know if you use any products जिसके ऊपर beeswax लिखा होता है. Have you all seen? Especially the infants, उनके soap में, उनके oils में ये beeswax होता है. It gives a very smooth and oily surface. Yeah, exactly. Beeswax is very famous. Okay. Waxes form water insoluble coating on hair and skin in animal. Waxes form an outer coating of stem, leaves and fruits. So we know cuticle, right? Then we know something called as suberin. All of these are going to be rich with fats. Yeah, look at this triglyceride beta. This is your glycerol molecule with 3 fatty acids. 3 fatty acid chains. Okay. Just give me 5 more minutes and we'll complete with the lipid part today. I won't go ahead with anything else. Quickly we'll complete the lipid part. Compound lipids, these are esters of fatty acids containing other groups like phosphate. So phospholipid, glycolipid. ये तो easy है ना? हाँ? एक लिपिड एक नॉन लिपिड. जैसे हमने देखा था प्रोटीन्स में भी एक प्रोटीन एक नॉन प्रोटीनेशियस पार्ट. In the same way, lipid and non-lipid. So they contain molecule of glycerol, 2 molecules of fatty acid and a phosphate group or a simple sugar. Some phospholipids such as lecithin have a nitrogenous compound attached to the phosphate group. Phospholipids have both hydrophilic polar groups and hydrophobic non-polar groups. So you know our cell membrane in cell unit of life. Same thing. Phospholipids contribute in the formation of cell membranes. Glycolipids contain glycerol, fatty acids, simple sugars like galactose. These are also called as cerebrosides. Large amounts of the cerebrosides have been found in brain. इसीलिए cerebral word से cerebrosides ये नाम आया है. In brain white matter and in myelin sheath. Just now I told you ना, myelin sheath lipids. Correct? Yes. Sterols. These are the last among the types of lipids. One was simple lipid, other was compound lipid, and these are the derived lipids. So sterols come under derived lipids. I am not talking about steroids. I am talking about sterols. There is a difference between steroids and sterols. Okay. They are composed of fused hydrocarbon rings. So the rings will be having what? Steroid nucleus and a long hydrocarbon side chain. One of the most common sterol is cholesterol. अब sterol बोलते ही दिमाग में सबसे पहले cholesterol आना चाहिए. No ideas. The one which is going to be produced naturally by our bodies are always good cholesterols or good sterols. Those which are consumed by us for no reason are bad for our health. ठीक है? So the common sterol is cholesterol. It is widely distributed in all cells of the animal body, but particularly in the nervous tissue. So see, your nervous tissue requires fats. So don't go on a fad diet where you are not allowed to eat even a little bit of fats. It's going to harm your nervous system. Okay? Cholesterol exists either free or as cholesterol esters. Adenocorticoids, sex hormones like progesterone, testosterone, vitamin D are synthesized from cholesterol. So cholesterol is not bad. The one which is going to be synthesized in our body, that cholesterol is required. अगर हमारे बॉडी में खुद ने गुड कोलेस्ट्रॉल नहीं बनाया तो ना एडिनोकॉर्टिकॉइड्स बनेंगे. एंड यू ऑलरेडी नो केमिकल कोऑर्डिनेशन चैप्टर में हमने देखा है कि अगर एडिनोकॉर्टिकॉइड्स नहीं बनते हैं तो व्हाट आर द इल इफेक्ट्स. करेक्ट. सिमिलरली इवन द सेक्स हॉर्मोन्स equally important. Correct. Cholesterol is not found in plants. In plants, sterols exist chiefly as phytosterols, that is plant sterols. Yam plant Dioscorea produces a steroid compound called as diosgenin. याम खाते हो? याम जो होता है ना Dioscorea का, it's actually what we eat is a modified stem of Dioscorea. Okay. It is eaten generally during fasting. It is not your ipomoya potatoes. Rataru nahi hai wo. It's not sweet potato. It's yam. Appears like that only. This particular thing, this diosgenin जो है ना, it is used as an anti-fertility pills. That is, they are used as birth control pills. Okay. So these sterols are also important. These are derived lipids. This is the overall, what we say, a summary of carbohydrates, sugars. ये देखो बेटा, ये मैं बता रही थी OH, OH दोनों जगह पर है तो ये राइबोस शुगर है. These are your amino acids. This is your lecithin, glycerol, triglyceride. This is your cholesterol molecule, adenine, adenosine, adenylate acid. So this is your double string structure of adenine, which will get attached with the sugar to form adenosine. So adenine plus sugar is adenosine, and adenosine plus sugar plus phosphate is adenylic acid. Okay, so it's just a summary. Producing these good cholesterol, yeah, our body requires good fats. Okay, good lipids should be consumed by us for making our body competent to make good cholesterol. And that, yeah, good fats मतलब कौन से? Almond oils. So we can consume almond, walnut. It's really good for our health. Okay, there are certain जैसे कि olive seeds, avocados. These are very good for even fish. Those who eat, those who consume good amount of fish. I'm saying fish. I'm not talking about meat. I'm talking about fish. Okay, certain fish also give us good amount of good fats, I would say. Okay. Of course. So yes, these all are going to help in giving us the chance to make good cholesterol. Well, so in the next class we'll go ahead with the dynamic state of body constituents, that is how metabolism is going to be functional in our body, and what are the important chemicals that are going to be produced in our body for this particular metabolism. Vaishnavi, I know you. Yeah, fine. So next class we can go ahead with completion of this particular concept of metabolism. Okay, fine. That's all for today, my dears. See you next week with continuation of this chapter. Okay.